So in EaSeq here we have 3 different types of data. They are presented up here in these 3 panels. They are called datasets, gene sets, and region sets. The datasets are your ChIP-seq experiments. They can also be RNA-seq, RIP-seq, methylation data, or something different. But they need to be mapped, so they need to be aligned to a reference genome before you can import it. And they also need to be single-read data. Then we have some gene sets, which are information about the positions of your genes and gene features in your reference genome. You can download it directly from UCSC using this download link. You just select the gene set that you'd like to download. And finally, we have some region sets, which are your points of interest, your regions of interest. Think of them as your viewpoints. You can so if you want to visualize, for instance, your ChIP-seq signal at CPG islands, then you need to have the coordinates of these CPG islands imported as a region set. They could also be enhancers or peaks generated with another program or from a published paper. And you can also use EaSeq to generate region sets. We will get back to that in a later presentation. Finally, we have an area here with plots. As right now it's completely empty, but as we go on, analyze and visualize the data, then this area tends to be filled with a lot of different plots. We will see more of that later. We have some tools controlling the plots, and as you can see, most of them are inactivated. They will require you to import some data. They suggest which type of data you need before you can use them. And we also have some tools here, some analysis tools, and they will also tell you what type of data they will need to become activated. So as we import different data into EaSeq, then you will see these icons becoming available. But before we do that, I'll just introduce the last panels over here. We have the Squeaks, which is a chat forum that you can use to write messages to other EaSeq users, and hopefully get some response if you turn into a problem. There's also another panel here. Right now it's not active, but if you click on a set of data anywhere in EaSeq, then it will show you some information about how that was generated and what was done to the data during your work or other people's work. So this will contain auto-generated descriptions of all the files or plots imported in EaSeq. And the final panel here is the navigation panel, where if you use EaSeq as a genome browser, then you can type in coordinates here or zoom in, zoom out. You will do that later on. If you think they take up too much space, then you can just minimize the panels, then they are not interfering with your work.